Dear students, this is our 20th lecture on medicinal chemistry and 6th lecture on antiviral agents. This is a special lecture on chemistry of COVID-19 wherein we will see that what is the structure of the coronavirus and what is the mechanism of its action in our body. Now, as we all know that corona disease uh, is a contagious disease caused by coronavirus SARS-CoV-2, and the first case for this disease was identified in Wuhan, China in December 2019. And the extent of this disease was such a huge impact on our life that WHO ultimately declared it as a pandemic. And till date, uh, in July 24, more than 7 million people have lost their lives due to this disease or pandemic. And it is believed that SARS-CoV-2 virus entered our body or human population through natural zoonosis. Zoonosis is basically a disease uh, where the infection of the humans is caused by a pathogen, and that pathogen may be infectious agents such as bacterium, virus, parasite, or prion. And this infectious disease can jump from humans to non-humans and from non-humans to humans. And the social and environmental factors including climate change, natural ecosystem destruction, and wildlife trade, which has increased the likelihood of such type of diseases which are spread over the globe. As far as the symptoms of COVID-19 are concerned, we have experienced that these symptoms were very variable depending upon the type of variant which we contracted, ranging from mild symptoms to potentially fatal illness. And The kind of common symptoms which were observed during this disease, they were cough, fever, loss of smell or taste. Then less common symptoms, they were headache, nasal congestion, runny nose, muscular pain, sore throat, diarrhea, eye irritation, and then either swelling or turning purple of our toes. Whereas in moderate to severe cases, the breathing difficulties were also observed in many of the patients. So, based on all these common, less common, and moderate to severe symptoms, the common cluster of symptoms have been identified into three categories. The first category is which will affect our respiratory system. So these are called as respiratory system cluster, where the symptoms are cough, sputum, shortness of breath, and fever. Whereas the second category is musculoskeletal symptom cluster, where the people will experience muscle pain, joint pain, headache, and fatigue in their body. And third category of symptoms is with the digestive system, that is digestive symptom cluster, where we will experience abdominal pain, vomiting, and diarrhea. So these are some of the common symptoms like sore throat, aches, pains, diarrhea, rashes on the skin, redness or irritation of eyes, headache. So these were many patients they have been found to have these types of symptoms, which were found very common in all those cases of the order of 88% of symptomatic cases. Now, there are many methods which are available for testing COVID-19, but the most effective and the common methods are these three methods. One is RT-PCR, real-time reverse transcription polymerase chain reaction. Second is transcription mediated amplification, which is called as TMA test. Then RT-LAMP, reverse transcription loop mediated isothermal amplification. So these are the three most effective and most commonly used methods for the detection of COVID-19. In RT-PCR test, the PCR technology was used to amplify small amounts of RNA from specimens into deoxyribonucleic acid, that is a small amount of RNA. They were transformed into DNA, which were observed by PCR technology. And this method replicated until SARS-CoV-2 was detectable if present in the body. So this test has been the gold standard test for diagnosing this COVID-19, and it was authorized for use in February 2020. It is the most accurate and reliable method for detection of COVID-19. Similarly, TMA mimics the retroviral replication, and unlike PCR, it is able to directly use RNA as well as DNA as the template, whereas here we are to use RNA as the template. So TMA is isothermic, so does not require thermal cycling device for detection of COVID-19. Then third category RT-LAMP, that is when a sample is taken from the nasopharyngeal swab. So this method is also of high accuracy for detection of severe acute respiratory symptom coronavirus 2 in saliva and nasopharyngeal or oropharyngeal swabs from asymptomatic and symptomatic individuals. Now, during those times, several methods they were used for the prevention of COVID-19, and now many vaccines have also been approved in various countries, and uh, many countries they have initiated mass vaccination campaigns for the COVID-19. In addition to vaccine, other preventive measures they were also used, like physical or social distancing, quarantining, then ventilation of indoor spaces, then use of face masks and coverings in public uh, to avoid coughs and sneezes, then hand washing, then keeping unwashed hands away from the face. So all these types of preventive measures they were used to avoid COVID-19. Uh, and some drugs have been developed to inhibit the virus. Although the new drugs have not been developed, only the other antiviral drugs or which are already used for HIV or influenza, they were tested for their activity against the COVID-19. Now, as far as chemistry of this COVID-19 virus is concerned, the TEM image, that is transmission electron microscope image of SARS-CoV-2 inside a cell, displays that it is a blue colored spherical viral particles which are due to this COVID-19. And the basic structure of this virus is that it consists of three basic building blocks, where we are having a single standard RNA genome. This is RNA genome, which is covered with a viral membrane composed of lipid bilayer, and this lipid bilayer is having surface proteins present on it. So the RNA genome encodes four types of structural proteins. One is nucleocapsid protein, and this is the nucleoprotein or nucleocapsid protein, which is present around the RNA strand. Then membrane protein is present in the lipid bilayer. So this is our membrane protein M, followed by envelope protein A. So these type of proteins, they are called as envelope proteins, and then spike proteins. These types of proteins, they are present on the lipid bilayer. They are called as spike proteins. And in addition to these structural proteins, there are many non-structural proteins present in the virus. Now, as far as nucleocapsid and protein is concerned, this one, it is a multi-purpose protein, and this protein helps in formation of nucleocapsid to protect the RNA genome. So this is mainly used for the protection of RNA genome, and the nucleocapsid is formed by packaging the viral genome into ribonucleoprotein complex. Then M protein, this one, M protein, this is most copious in viral surface, and its key role is to support the viral assembly as central organizer due to its membrane binding properties. So this helps in viral assembly. Then E protein, E protein is sorry. E protein is the smallest membrane protein, and it comprises of 76 to 109 amino acid residues, and it has a crucial role in viral assembly, in valve formation, and membrane permeability to the permeability of the host cell and the virus. So this helps in attachment of the viral virus to the host cell. Then is S protein or spike protein. Spike protein, this is an important structural transmembrane protein, and the number of amino acid present is very high, of the order of 1200 to 1400 amino acid residues. They are present on the outer envelope of the virus, and this protein or spike glycoprotein is responsible for the entry of the virus into the human cell, because this recognizes the specific host receptor located on the human cell surface. So this protein will interact with the receptors of the human cell, and then there will be binding of this protein with the host cell receptors. Then host guest recognition is virus specific, and this specificity and decides both virus tropism and pathogenesis in case of human cell surface and the virus. And this S protein exists as a self-assembled homotrimer. In each of the monomer is composed of two functional units. So this S protein has two subunits. We can say that S1 and S2 subunits. S1 subunit is accountable for host recognition, where S2 subunit is in charge of the host guest membrane fusion. So S2 subunit will be used up in making a fusion of the virus with the host cell. Here, the N-terminal domain of S1 subunit, uh, this one, this is considered as carbohydrate recognition domain, and C-terminal is called as receptor binding domain. So both the N part and the carbon part of the amino acid in case of S1 subunit will be used for different purposes. So one for carbohydrate recognition and other for receptor binding. And this receptor binding is particularly responsible for virus and entry by recognizing protein receptors of the infected lung cells. Now, how this virus acts in the human body, or how it enters, and then what is the mechanism of action for the replication process of this virus in the body. So basically, viral replications cycle or life cycle of this virus starts with attachment of this virus to the host's human cell. So this fusion, as I've told you, take place with the help of protein which is present in the virus, and then fusion of the viral particle with the host cell membrane is followed by entry of the virus inside the host cell, where after which the uncoating of this viral membrane take place by endocytosis. Uncoating take place, and after uncoating, the viral nucleic acid mRNA is released in the cell as viral genomic RNA. Then translation of this synthesizes polypeptide chains, and then proteolysis of these polypeptide chains gives us fragmented proteins, which through RNA replication are converted into functional proteins, and these functional proteins through translation are converted into structured proteins of the virus, and then assembly of these structured proteins lead to through packaging again forms a infected virus, and this through budding is released to the body. So this is how an infected virus is released in the body of the human. Now, as I've told you that the host receptors they interact with the S protein, which facilitates the virus entry into the cell, and most effective fusion take place with these ACE2 receptor and TMPRSS2 receptors. So through these two receptors present on the human cell, the virus gets fused to the human cell, and ultimately this passes through the cell wall into the cell. Here, during the entry of the virus into the cell, two large polypeptides, PP1A and PP1AB, they translated from the viral genome single stranded RNA. They are released into cytoplasm, where they will break and mature into non-structured proteins, as I have told you. And these non-structured proteins or fragmented proteins or functional proteins, they undergo RNA replication, which is facilitated by viral replication complex, which also includes helicase or RNA-dependent RNA polymerase and other auxiliary non-structure proteins. Then these, through translation, are converted into structured proteins like M protein, S protein, and E protein. They are produced in the cytoplasm and subsequently transferred to the Golgi intermediates, where the endoplasmic reticulum interacts with the structured proteins, and then a virus infected cell produces a large number of these building parts. Then this will undergo self assembly to form new viruses. And finally, these new viruses which have been formed, they are exported from the infected cell through a process called as exocytosis. Cytosis. The Then the virus has a spherical supramolecular structure in which all the building blocks, they are associated by weak non-covalent interactions. And because these interactions, they are very weak and non-covalent in nature, hence very mild chemicals such as soap and detergents, they are enough to split the unit and destroy the virus. So this we have seen in our during our cases of infections, that by washing with soap or detergent, the virus can be destroyed. Now, as we have seen the different steps of the replication cycle of the COVID virus, therefore, based on these various steps, it was thought to develop antiviral drugs. So, most common antiviral drugs, they were used for each of the step where the changes in the virus is taking place, either before the entry into the cell or after the entry into the cell, when different steps, they are being taken place. So first step, as we know, is the entry of the virus and then fusion of this virus with the human cell. So to inhibit the activity of this entry and fusion or endocytosis, few drugs, they have been found to be very effective, and these drugs, they are called as entry inhibitors or fusion inhibitors or endocytosis inhibitors. And most common among these is Abridol, then Camostat mesylate or chloropromazine. These are the three most commonly used drugs which are effective to inhibit the entry of the virus into the cell, or fusion of the virus to the cell, or the process of endocytosis of the virus. Then some endosomal maturation inhibitors, they were also used, which inhibited the uncoating process of the virus in the cell. And the most effective among the drugs is hydroxychloroquine. Hydroxychloroquine was useful that in it inhibited the process of uncoating, so this acted as endosomal maturation inhibitor. Similarly, conversion of these polypeptide chains into fragmented proteins, this can be inhibited by protease inhibitors like lopinavir or tenofovir combination, or atazanavir, darunavir, or secunavir. All these antiviral agents, which are used for some other purposes like HIV or influenza, they were also found effective for inhibition of this process of proteolysis. Then one more drug which was used for inhibition of RNA replication activity. So RNA polymerase inhibitor. These are the most effective drugs. Emtricitabine, azuvidine, favipiravir, and remdesivir. All these they acted as RNA polymerase inhibitors, and out of these, remdesivir was most commonly used and most effective as nucleoside or nucleotide analogs to inhibit the viral genome replication in the COVID-19. And similarly, dexamethasone was used as an anti-inflammatory drug to control the immune system in general for all the COVID-19 patients. Then oseltamivir, Tamiflu drug was used as neuroamidase inhibitor. So it inhibited the exocytosis process of the virus. So these drugs which were developed for some other antiviral activities, when they were tested against COVID-19, they were found effective for the patients of COVID-19. So these are the references which have been used in preparing this lecture. Thank you very much.